Yep.
We made a flight payment to Tennessee.
Wait, what?
Oh my god, today's Friday already?
Dang.
Oh my god, this is a last class.
So tomorrow, I have something interesting to do. Apparently, I'm getting inducted into the National Honor Society.
Ooh, congrats. Very nice.
Yeah, I wasn't planning on going, but my mom said you're going.
Yeah. My mom would do that too.
She got tickets and everything, and I was like, okay. I didn't even know I was getting invited.
He approached me and he said, "You haven't done this in Preston's lecture yet." So, do you have any idea when we're going to be doing?
What are you covering?
We're doing that corn genetics lab. It's
Yeah,
where you
corn.
go in and you look at the phenotypes.
I don't know what I was doing.
Okay.
It'd be coming back that usually the last section of this class.
All right.
And, I would say that will be, we'll probably get started on the
I made a
weekend.
mistake starting the video already.
No, no.
I made a mistake starting the video already.
And,
Oh yeah.
after you take this test on Wednesday,
Dr. Malik was saying I should maybe look ahead if possible to be prepared for what's
I
coming.
can put them up, you know.
Why is he talking about this right now?
I can put it up, as soon as, if I get the chance to today, I can go ahead and put that unit up, yeah.
I just,
It's in your book, but
I
it's
have the
on,
same
it's kind of
professor
spread out.
in the lab, but I don't know how to do the corn. I was just like, I'll just wait until I learn.
Yeah, me too. We're doing the same one for Dr. Stone.
We're at the micro
just wait.
spindle or placebo.
I just wish I got the same person for each thing.
Yeah. Yeah. Honestly, I'm hoping for that next semester.
So I'm just gonna ask, since you plan to do biology too, would you mind sending me who you're doing? Because I'd like to have at least somebody to know in there.
If it fits with your schedule, yeah. So it's Monday, Wednesday mornings.
Yeah, I literally have nothing to do.
Yeah, normally I take afternoon labs, but I was like, I'll try morning. Whatever. I think there's lots of dissections and things. So I'm excited about it.
Haven't dissected a thing yet.
Oh, Dr. Preston,
I've
is there
dissected a
anything
rat
that we
before.
need to draw up to this
I
point?
think I might have had to dissect a frog once.
'Cause I know I
like in
haven't heard
junior
anything.
high.
Yeah.
And that's about it.
I don't know. I can't remember. I think I did a frog too, but I definitely did a rat, and it smelled so disgusting. It was so gross. It smelled. They soak it in a
Guys, just so
preservative.
everybody knows, we will finish up this unit today. And Monday, I will do a thorough review of what's gonna be on the test Wednesday. As I was telling Adriana earlier, concentrate on the chapter on photosynthesis and meiosis. There's not gonna be that much over the chapter 11 that you see there. There'll be some of the things I told you about, basic stuff, but don't go into it very deep. And you'll know exactly on Monday. Most of the questions are going to cover photosynthesis and mitosis, which is the cell cycle here. If we look at these, we had gone through, I'm going to bring this part back up so that we could look a little bit at this part. Let's review with mitosis and cell cycle. Two kinds of cells in your body. The gametes, which are the egg or the sperm or the sex cells. Everything else is called a somatic cell. Also remember, when we get into cell division, you really, guys, have to pay attention to the terminology here. What are the two domains of cells in general? And those would be prokaryotes and eukaryotes. Prokaryotes being only bacteria. And eukaryotes, everything else. The two domains. The two types of cells would then be either somatic or gametes. They can be broken up, obviously, into different groups. So it's kind of like a pyramid building up. Here, we are talking about the formation of regular somatic cells. Mitosis is what gives us a somatic cell. Meiosis or mitosis then gives us the reproductive cells, so a sperm. Big difference. Somatic cells that we're looking at, when they are finished, will have a diploid chromosome number, meaning they will have two sets of chromosomes, one set from the mother, one set from the father. Sex cells or gametes will be haploid. They will have only one set from each, 23 chromosomes. So also remember, whenever you are asked what is the chromosome number of an animal, they're talking about how many sets does it have, not how many actual chromosomes. Does it have a one set, two sets, three sets? Most mammals, most plants and things like that are diploid, two sets, one from the mother, one from the father. Sex cells have one set, haploid. Some plants and some of the lower maybe versions of animals could possibly get what is called a triploid numbers, where they would have three sets of chromosomes. So this talking about right here is how do we get that diploid number for a somatic cell? Two full sets, one from your mother, one from your father. You will see when we start to talk about heredity that you are going to have a couple of genes in there. Let's say for hair color. With those two genes, you can get, one of those is going to be expressed, those genes. The one that's expressed will be said to be the dominant trait. The one that is there but not expressed is called the recessive trait. Dominant traits always show up if they are present in most cases. You'll find that it's not as simple as that. It can get quite complicated, but that's the basic theory. So if you've got brown hair and black hair parents, and they give you one gene or allele for that brown hair trait, one gives you the black hair trait, which one shows up? There's not anything that really determines which one shows up. It's which one will be expressed. Now, which one will be expressed, the only way we can do it is look, see which one actually does show up. Then we might be able to say black hair is dominant over brown. Or we might take a different situation and say brown hair is dominant over blonde, or blonde's dominant over whatever, however it shows up. And this is all done randomly. It is not anything that guides which trait that you're going to get. Nothing guides which one's going to show up. You've got a small pool of possibilities, and just whichever one luck happens to give you, that's it. It's not guided. And as we said, you don't look half like your mother and half like your father. Scientifically, if you think about it, that's exactly what you should look like, right? Mm-hmm. You've got one set of genes from one set from the other, so you should look half and half, but you don't. Some people look a lot more like one parent than the other. Sometimes they don't look like either. That's because when those genes come together, they can change the traits. That is what you have heard scientifically done in a genetics lab called recombinant DNA, but it happens in your body. It does it naturally. It gives you different combinations, so you don't look exactly like that. This is the opposite. This is just copying exactly what you have. It's changing nothing. It's just like if you took this sheet of paper and ran it through a copy machine, you will get out exactly what you put in. That is what mitosis does. If you have a skin cell that is injured, it is going to copy that skin cell, and it's going to have the same exact genetics of what was there before. just 2 new versions of it, 2 copies. That's what somatic cells, mitosis, the cell cycle gives us. When we look at that, we said the big part of mitosis, one of the things that I want you to remember here, right there. There are parts to the cell cycle. This whole thing represents the cell cycle. The cell cycle is divided into 3 parts. Again, pay attention to this for the terminology whenever you take the test, so that you can understand the way the question is asked. There are so many different things that we have to give specific terminology to these to denote which one we're talking about, and you need to be aware of that. If I ask you, what are the parts of the cell cycle, you should answer interphase, mitosis, and cytokinesis. Those are the 3 parts of the cell cycle. If I ask you what are the parts of interphase or stages, it would be G1, S, and G2. Remember, in G1, all of the parts of those 2 new cells that are going to come about, those parts are being made in G1. In the S phase, that is where the DNA will be copied. The synthesis phase, it will make copies. In the G2 phase, that is where the cells will mature and we start to get chromatin and so forth put on the DNA. So, at the end of interphase, we should have the parts to make 2 new cells, and we should have the DNA turned into chromosomes and copied. Then we start mitosis. The first stage of mitosis, we'll go back to the other picture where we had. This is where we're coming out of interphase. Notice, now we've got the chromatin over the DNA at that stage. So this is the end of the interphase, and then we start mitosis with prophase. With prophase, chromosomes start to become structured and organized. When it says the word organized, it means the chromosome pairs that were copied are going to match up, and they're going to come to certain places within the cell separate from the other chromosomes. You can see that starting to happen here. They have not completely structured, they have not completely separated, but they're starting. This is prometaphase. Notice here, in prophase, we get these 2 little components called the asters. These are kind of, think of them as an anchor on the top and bottom of that cell. To them will be attached these fibers that you see, spindle fibers. They will attach to the centromere where those 2 sister chromatids, now we had 1 chromosome, we copied it, it's held together by the centromere. Then there will be a spindle attached to each chromosome. That's in the prometaphase. And they are, as you can see, starting to become more spread out and more separated. Next, we had prophase, prometaphase, where they started to get structured, now metaphase. And if you look at metaphase, now they are organized on the equator of that cell, right through the equator part. That's in the middle of the cell. Each one of these has a spindle fiber attached to that aster, and they are now going, those spindle fibers are going to start to shorten. And as they shorten, they will start to pull chromosomes towards the poles. Here in anaphase, you can see they have separated from their sister chromatid. They are no longer, here they're a pair of sister chromatids. Here, they are now not chromatids, they're chromosomes in anaphase. They have separated. Each of these will travel towards the asters, towards the poles. A nuclear membrane will start to form. So what makes the nuclear membrane, did we say, or internal membranes?
The ER, the
ER. So we've got the organelles in place to do what the cell needs to do. So now that we've got separation and they move to the asters, we start to get this purplish color in through here is the nuclear envelope. Now you can see the nucleus starts to form. It will eventually bring all of that genetic material. It will be enclosed in the nucleus. And we will start to see these cells. Here in anaphase, they're starting to elongate, like if you had a round little sponge ball and you started to pull it, it starts to get oblong shape. And that's what this is getting as opposed to round. It will eventually, sorry about that. I had power lines come down and I had to turn my phone on for Duke Energy and everybody. But they will start to pull apart and eventually they will come all the way apart. Where they start to come apart is called the cleavage furrow. Remember cleavage, and I might ask you this on the exam as a definition. Cleavage means to cut. Think of a meat cleaver. So here we have these start to pull apart. Notice, they do not separate telophase and cytokinesis. The reason is there is not a specific place where telophase stops and cytokinesis begins. So they often include those two together. At the end of cytokinesis, these cells will not be joined together. They will be apart. That is mitosis. So what we have done, we started with one cell, copied everything in that cell, the genetics and all, and then just made two cells out of it. Now let me ask you this. What happens if there's a defect in the genetic makeup of that cell that's being copied? Go for it.
Is that copied also?
Absolutely. So you can have some cells that could have some kind of condition, diseased state, something abnormal, and that will be passed along too because it's just copying the DNA. That's all it's doing once there. And cancer is something that people sometimes have a confusion with. Cancer itself is not a disease. It is a bunch of diseases called cancer. They work along the same way, but they can come from different cells. They can have different causes, but they come from different tissues. Cancer cells can be caused because you've got a genetic defect in there that says you're going to get it. It could be because of environmental conditions like radiation, asbestos, things like that. That can be a cause. And it can be just chance mutation. In your bodies, it has been estimated at one time that every person will get cancer 14 times in their life. So why don't you get cancer? All of you in here have had it already. Why do you not have it?
Is it because the defected cell is destroyed?
Your immune system is powerful enough to locate them and destroy them before they can go through this process and grow. So why do some people get cancer then and others don't?
Immunodeficiency?
Why do younger people have younger and older people have a tendency to get it more than the middle age and system health? As you start to get older, your immune system starts to go down, so you're more susceptible to it. And if everybody lived long enough, you would all die of cancer before some other condition got you. But as long as your immune system is strong, it can keep knocking those down. Mutations happen normally. I mean, they occur. Some of them slip through the system and can grow. Now, what cancer cells do, this process of mitosis, whenever that goes through, as we will find when you take cell biology, there will be chemical signals sent to the cell saying, stop mitosis. Now, go through it once, but that cell, stop. Cancer cells have a way of making that malfunction. Cancer is the overgrowth of cells. That's what it is. Uncontrolled growth. And eventually, they will grow into so many cells that they will make either tumors or whatever. And cancer, one of the things it does is usually mimics whatever the function of the cells that it came from. If you had an adrenal tumor on the adrenal glands, it will start to get to the point where it will actually put out way too many of the hormones that the adrenal glands would normally put out. So cancer basically does the body's system control when these cells start and when they stop. Yeah.
Now, what causes, so this is a case that I'm wondering too about the tumor formation. And so my cousin, back when he was like 10 or 11, he had an undifferentiated embryonal sarcoma of the liver, you know what that is? And the thing actually like burst. How is that caused by the dividing of the cells as well?
Overgrowth, yeah. Simply overgrowth. The difference between cancer and a tumor, I probably shouldn't tell you this, but to confuse you. Medically, a tumor is always non-cancerous, benign. Cancerous cancer is a malignancy. Even doctors now will say a cancerous tumor. That's technically not true. But the difference is one of the ways within your body, and I can give you an example of one thing that occurred. If you have a tumor, it will be walled off with a membrane around it. Think of if you had a rubber ball and you had a tough membrane around it, separated it from wherever else it was at. Cancer does not have that membrane that separates it from the rest of the body. It actually grows into the other tissue. I had one guy come to me one time who had a big knot in his throat. He'd been to surgeons and medical doctors and all kinds of people. And they say, well, you've got a problem probably either with the gland or with the tooth. Well, he didn't. And one of the things you do when you see that, you stick a big needle in it. And the purpose is make sure there's no blood. You can have little tumors made of just blood vessels, hemangiomas, that you go into them and they will bleed to death on you right there because it's such a network of blood. Trying to get the arteries shut off is difficult. So put a needle in it, nothing came out, no fluids, nothing. And keep in mind, this is about the size of a golf ball down in your throat. And so I said, okay, took a probe, which is a sharp needle basically, and just went down into it, nothing. It was like trying to stick it into a rubber ball. Well, the guy had been passed around so much, he just kind of raised up and he said, fine, do something. We will. So then on thing to do. Took a scalpel, went in, opened it all up. And when you opened it up, moved all the tissue back away, it kind of looked like a little gray rubber ball. But when you cut open on the inside of it, it was solid. It was attached all through there, and it looked like really old cottage cheese, gray-green cottage cheese was the consistency. And when I done that, I knew what it was. Sewed it back up. It said he told the surgeon, you better look again. And he had the type of cancer at that time. This has been some years back, but at that time he just didn't treat it. It's called pleomorphic adenoma. And, you know, you look at him and you say, this guy's done. You know, there's nothing to do. But that, you could tell it was growing. It wasn't something, if you've got a bump on your skin and it moves around and things like that, that is not anything cancerous. If you got a big one that grows and you can't move it, it could be. Doesn't mean it is, but it could be. So, most doctors, when you look at those things, you can tell straight out pretty much what it is. So, but it all comes from these cells. If those cells continue to grow and grow down into other tissue, you got a problem. One of the reasons they tell you, if you think you got something wrong, always to make sure that you get it checked out, take the skin. If you start getting a black spot on the skin, that could be melanoma. If it's on the skin, you can handle it. Not a problem. But if it keeps growing and grows all the way down into the tissue where it's not movable, then it has gone from being in a place that doesn't have vascular passages to the other parts of your body where it can metastasize. If it gets down in connective tissue, then it spreads and it becomes extremely difficult. So one of the things, the stages of cancer is how far does it spread into other tissue? And that's why doctors always say, and you're always warned, if you think something's wrong, get it checked early. Because usually, you got something like a melanoma, cut it out right there in the office. And, you make sure you cut down below it so there's nothing left, that you get it off. A lot of times you don't have to do anything. It starts getting down to the tissue, then you're going into chemotherapy and other things trying to kill it throughout the body. So this stuff is important right here. And it is the basis for many diseases. Think about this. What happens if this doesn't occur? One of the things,
No.
you don't have healing, right? So, if you don't have healing and you've got tissue that was hurt, that's a problem too, right? These are genetics, but the thing, two things to remember. You have to stop this cycle and start it. And your body has to be in control. And whatever the genetic information is in there will be the genetic information that is then going to be changed. So these could be difficult. And if you guys ever go through to most of the medical professions and things like that, you'll find out most doctors, they will never tell you what they think, but most of them know when they look. They won't say anything until they know for sure. And sometimes you can't know for sure, but sometimes they think it definitely is and it's not. You don't want to tell somebody they might have something like that. But cancer treatment cures, folks, is enormously improved. There were things that if you had gotten 20 or 30 years ago, you wouldn't have made it. Now they treat it and people do fine with it. And probably by the time, in another 20 years, it will be very possible that when you get cancer, you just go to the doctor and you get a vaccine, you go back home. You're done. They're working on vaccines right now that will target only the cancer cells, kill only the cancer cells. And we've got plenty of ways to kill it right now. You just don't want to kill every other cell in the process. So it's how do you target these? That's where those receptors, like in chapter 11, come in. If you can target to kill only the cancer cells, we've got stuff right now that'll do that. So, that, mitosis is important. It's a big deal. And, I mean, it'll happen to everybody within families somewhere. Most people will have somebody that could have some kind of a problem with that, whether it's something like malignancy or other diseases. These are called metabolic diseases. Cancer is called a metabolic disease because it involves cell replication and all of that. Now, we went over most of those. Let's look at these again. This is going to explain. We had started with, the mitotic spindle, structure made of microtubules that controls chromosome movement. That's those little yellow fibers that we were talking about. And remember, these little concentrated areas are called asters. That's where the spindles are attached to. In animal cells, the assembly of spindle microtubules begins in the centrosome. The centrosome replicates during interphase, forming two centrosomes that just migrate to the opposite ends of the cells. These asters are where all these little microtubules come together and hold them together. Remember, in the middle of these chromosomes, after they've been duplicated, there will be one little spot where they're connected. That is called the centromere. Spindles will attach to the centromere on each chromosome and then pull them apart. The spindles will shorten and pull them towards the asters. It says during metaphase, some spindle microtubules attach to the kinetochores of the chromosomes and begin to move the chromosomes. Kinetochores are protein complexes associated with the centromeres. In other words, there are little parts of the centromere where, on each chromosome, where the spindle can attach to. And then we talked they were pulled towards that equator of the cell called the metaphase plate. I might ask you on the test, what is the name of the equator line where the chromosomes, sister chromatids are pulled to? When I say sister chromatids, what am I saying? What does that tell you about chromosomes? Sister chromatids.
That's when they're actually in the X form.
They're attached.
They're attached.
Whenever they are pulled apart, then they will be referred to as a chromosome. So remember, chromatids means they are just two chromosomes that's attached. When they are pulled apart, they become chromosomes. And this shows the process once again. Asters. The whole thing is an aster. These are where these little fibers come up. Spindle fibers will then attach to the kinetochores on the chromatids and start to pull towards the poles to separate.
So the aster, you're saying the whole thing's an aster, even the centrosome's considered an aster, or
It's
does?
part of it.
Okay, so the centrosome doesn't produce the aster, but it's, or, because I know that the centrosome produces the
The
microtubule.
centrosome is this little concentrated part in here,
Right.
and when you attach the spindle fibers to it, that makes the aster.
So there's
Okay.
a centrosome in the aster, and there's also one in the chromosome.
Centromere in the chromosome.
Oh.
Centrosome is the whole, it's the little anchor to where the spindle fibers attach to. When the spindle fibers, these ends are attached, like you see around there, then it is called an aster, the whole thing.
Ah.
So the aster makes up the centrosome and the spindle fibers attached to the centrosome. The other end of the centrosome attaches to the centromere, the part that holds the two chromatids together.
Okay.
What was the breeder called?
The what?
The breeder? What was it called?
metaphase plate. Remember, metaphase is where their chromatids are all pulled to the midline of the cell. Meta means metal. That's just middle phase plate or metaphase plate. And this shows an actual picture that you'll get to see whenever we do the lab. I think we do the lab on genetics Monday after Thanksgiving, so.
What's the difference between kinetochore microtubules and non-kinetochore?
Go back. From what you're seeing in here, see how these are coming this way? They're on the pole side of the ester. And these are overlapping through here. See this? It's not attached to the kinetochore. They're pulling more on other parts of the cell and not just chromosome. So the biggest thing to remember about that is the kinetochore spindles are attached to the centromere. Non-kinetochore are not attached to that part. And this part is just like what we talked about. The spindles start to shorten and pull the chromosomes in towards the poles. Here they are. Non-kinetochore microtubules from opposite poles overlap and push against each other, elongating the cell. They're not pulling on the chromosome. They're working on other parts of the cell. Think of it as this process occurs, which was a good question. We go back to look at this. See how this is starting to elongate? See right here? That's a non-kinetochore. It doesn't attach to the chromosomes. But it is still playing a part. These are shortening. These are elongating and pushing. That is what makes the cell start to push apart, elongate. Now, cytokinesis here. Remember, cytokinesis is where the cells start to separate, not the chromosomes, but the entire cell now is starting to separate. Membranes being formed, so we will wind up with two cells. When, at the end of cytokinesis, both cells will be completely apart, which would be another good question on this. When do the cells that are dividing come completely apart, separate? At the end of cytokinesis. It starts with a cut called cleavage. As that cut elongates or widens, it's called the cleavage furrow. But in plant cells, it's different. Plant cells have what that animal cells don't have. From junior high science. Cell wall. Cell walls are not going to be flexible. They're going to grow and they're going to be there. So at the end of cytokinesis, a plant cell will have two cells just like in animals, except it won't be in this elongation process. When the cell forms, it's going to be like a big block, and whenever that middle wall forms between the two, that's the cell plate. that makes two cells, as we will see here. This is what happens in an animal cell. Here, this is where the cell plate will form, and then become that new cell wall. So there's no, where you've got the non-kinetochores in animal cells that are pushing and elongating, pushing them apart. This cell is just formed the size it's going to be, and then gradually there will be a cell plate forming once time comes to separate. So in a plant cell, cytokinesis is going to mainly be involved with the development of the cell plate. And it's kind of interesting. This shows you what one of those cells will look like. We always draw these cells as flat little circles on the board. They're not. Think of them as like this ball right here. It actually just starts to turn in on itself. This will get larger and larger, and it will eventually just pinch apart to make the two cells.
I have a question.
Mm-hmm.
In G1, did we say that the parts of the cell are, like, all the organelles are reproduced? I mean, they're
Parts are being made, yes.
Okay.
How do we know what to make? Where does that information come from?
The genes.
DNA, like everything else. And so there will be a code for making those organelles, and just like there's a code for every other thing. This shows what you will be looking at a lot in the labs when you do the labs. This will show you different phases. Notice here, where you've got this big jumble is prophase.
I actually have some raw footage of that on my phone because I already did that lab. You want me to share it with you?
This is, look at that area. Notice what it's called. Nucleolus. Nucleolus is a condensed area inside of the nucleus. That is where the actual DNA is made or contained. Outside of that nucleolus, inside of the nucleus, is the nucleoplasm. The nucleoplasm is the gel-like fluid that's in there that will contain the things for the resources, basically, to make the copies, to make mRNA, to do things such as that. Notice here, prophase, it's just come out of interphase. It's just a jumble of chromosomes. You could even argue maybe this is late stage interphase. That's one of the things when you guys look at these on the slides, sometimes whenever the different phases are back to back, and one's ending and one's starting, it's hard to tell, is it ending or is it starting? It's hard to tell. That's kind of a judgment call. But this jumble right here is prophase. Notice now, if you look, they're starting to organize. Don't look as jumbled. Here, they're even more organized. And this is the way they will look. They are pulled towards that plate. And if you notice here, they're starting to separate in anaphase. How can you tell that's anaphase? Notice that these little V shapes, if we take this right here string and then pull this way in the center, then the string pulls, makes this little V as we pull it because the ends are loose. So that V basically points towards the pole that it's going to. And you can see some down here, some up there. Telophase, notice, in telophase, these have separated now, and we're starting to see separation. Now, let me ask you this, are we looking at a plant cell or an animal cell? You should be able to tell.
Plant cell.
How can you tell?
The cell wall.
Notice right there, we are getting the cell wall, and we're starting to get that cell plate formed. Starting to form, it's not there. You could also say, that's why, is this late telophase or is that early cytokinesis? Maybe both. There's still processes of telophase going on, and there are processes of cytokinesis that have started. About bacteria. Bacteria just basically, the cells, it copies the DNA like what we've talked about, and then just separates. It is a cell. It's not going to make, it doesn't have a lot of the other things cells do. This has got one cell, it's going to copy all the material, it's going to separate, make cell wall and separate. That is called, what I want you to remember, binary fission. Right here. We've got just a couple of little slides left on this. We will start with the control system of the eukaryote side. There's like 10 slides or so. And we'll finish with that Monday and do the full review and for the exam. There's not that